Good afternoon. Today is Sunday, June 7th, 2020. My name is Ricardo Sandoval, and this video is the results video for lab number 6, biochemical testing for microbial identification. This is for Biology 225, microbiology at Los Angeles Pacific University. So in this lab, what I did is I performed 3 different biochemical tests. I performed the methyl red test, the Vogt-Proskauer test, and the catalase test. I didn't really have issues, so to speak, with any of the tests. I did have to allow my microbes, my E. coli, and my Staphylococcus epidermidis to incubate for about 4 days. Incubation started on Wednesday, in the late morning of Wednesday, June 3rd, and concluded this morning. Just because after 2 days I didn't see much happening, and then, of course, life got in the way, so I did use the cultures today. So, the 3 tests I performed again were methyl red, Vogt-Proskauer, and catalase. So the methyl red test identifies microbes or bacteria that perform what's called a mixed acid fermentation. And this is what's defined as an anaerobic process. So anaerobic, of course, means no oxygen needed. And the process produces lactic acid, acetic acid, ethanol, carbon dioxide, and hydrogen gas. So we've learned about the pH scale. We have acidic, and then we have basic. And then on the pH scale, something on a lower number is acidic, something with a higher number is basic, and the pH scale can also be identified by color. So how this works is this uses a solution called methyl red that after a bacteria, in this case Escherichia coli and Staphylococcus epidermidis, are cultured, incubated in the MRVP broth for at least 2 days. I believe it was 12 drops of this methyl red solution go into both tubes, and if the solution turns red or pink, the test is positive, meaning it's acidic, that there's some sort of acid in there. And again, because the acidic parts is on the left side, and mixed acid fermentation does create an acidic solution, red is the color we're looking for. So these are my test tubes here from the test. Now, they've been sitting for several hours since I performed the test. So the photographs, my final reports, might be slightly different color, but for both E. coli and my S. epidermidis, the methyl red tests were positive. So mixed acid fermentation did take place in both. And again, no issues. Quite an easy test to perform, and quite enjoyable to see that both tests turned a really beautiful bright red almost immediately once the methyl red was added. So the next test I performed was the Vogt-Proskauer test. And this identifies microbes that ferment glucose, which is sugar, via what's called a butanediol pathway. And then this pathway produces a substance called acetylene, and an acetylene can be used for fuel. So this acetylene, in the presence of what's called a Barrett's reagents, A and B, turns red if the acetylene is there. If that butanediol pathway, a metabolic pathway for both E. coli and S. epidermidis takes place. So how this test took place is, again, I had test tubes with the MRVP broth with the S. epidermidis and the E. coli. I added both drops of the Barrett's reagent A and B, and the test tube sat for 30 minutes to see if I would get a positive or a negative result. So after 30 minutes, I saw a very faint, very diffuse red on the E. coli, while the S. epidermidis remained clear. Now, again, keep in mind that the test tubes I'm going to show you here have sat for several hours. So, instead of the top layer, I now have a bright red tube for my S. epidermidis, and for my E. coli, I should say. And the S. epidermidis is this kind of rosy color. So, I'm still going to stick on my things, and E. coli was positive, S. epidermidis was negative. You'll see the photographs in the final report that will be attached. Again, no issues performing this test, relatively straightforward. My cultures were fine. When I took the cultures out of my incubation location in my home office where I am now in our closets, there was some flocculence at the bottom, but no turbidity. And as we'll see in the experiment video, as I was pouring from the MRBP broth into these test tubes, quite interesting was that the initial pour ended up being turbid in the test tube, and then the second pour was clear, which is quite interesting. But the results did turn out as expected. And the third test that I performed is called the catalase test. And as I stated in my experiment video, the suffix ASE lets me know that catalase is an enzyme, and enzymes make things happen. They act as a catalyst. So in this case, what catalase does is it protects against the damaging or oxidative effects of H2O2 or hydrogen peroxide. So we all know hydrogen peroxide as an antiseptic. When you cut yourself or when you have a mouth sore, for example, you put some hydrogen peroxide on the wound or gargle some with some water, and what you see is fervent bubbling, and that lets us know that it's killing bacteria. So in this case, though, what we're looking to see is if catalase is present in both the E. coli and the S. epidermidis, because what catalase does is it protects against the damaging effects of hydrogen peroxide, because when we put it on a wound, it's killing bacteria, but from a microbiology standpoint, we want to allow certain bacteria to thrive, especially if it's normal flora. So in this case, what I did was I grabbed 2 slides, labeled them E. coli, S. epidermidis, drew a dime-sized circle with a Sharpie, and then took a sterilized inoculation loop to the petri dish for my E. coli, and then also for my S. epidermidis, and then rubbed it on the slide. After that, I then poured, using a pipette, I put 1 drop of hydrogen peroxide on each slide. And then what I was looking to see if catalase was present in each was fervent, immediate bubbling. So, again, the slides I have here have been sitting for several hours. But this is my E. coli, and you can see that kind of white bit in the center, and that was where it was bubbling. Kind of the entire black circle, actually. And then this is the S. epidermidis. There we go. So when I initially placed the hydrogen peroxide on the E. coli, the bubbling was immediate. So, and very fervent, very active. So I knew immediately that I had a positive catalase test with the E. coli. When it came to the S. epidermidis, I put the drop on and nothing really seemed to be happening, so I assumed that the result was going to be negative, that S. epidermidis did not have catalase, and that if hydrogen peroxide is placed on, it's just going to do its thing and kill the S. epidermidis. But after about maybe 10 to 30 seconds, somewhere in that range, I did notice that it started bubbling. And as you can see on this slide, that kind of dried portion, that's where the bubbling kind of stayed. So, interesting to know that S. epidermidis does have catalase, because S. epidermidis is normal flora, but for patients in the hospital, for example, again, I'm a nurse and I have a patient right now, well, 2 of them, that both have IVs inserted, and if ever there's any sort of issue with their skin integrity or the catheter or sterilization, S. epidermidis could develop and cause all sorts of problems. So again, no real issues with any of the labs. It was interesting to see how everything worked as far as color. That made it very vibrant. And again, mixed acid, of course, acid is acidic, so on the left side, the red was good. The Vogt Proskauer, the butanediol metabolic pathway, acetoin, it can be used for fuel, and it turns red when exposed to Barrett's reagents A and B, which after 30 minutes, what we're looking for is that red top layer, which I barely saw on the E. coli and didn't really see at all with the S. epidermidis. And then catalase itself protects against the harmful effects of hydrogen peroxide, which can kill. And then both microbes, the E. coli and the S. epidermidis, were positive for catalase, which means that both microbes, bacteria, won't immediately die, or won't die at all for that matter, when exposed to hydrogen peroxide. Because how catalase works is it separates the hydrogen atoms and the oxygen atoms, and the oxygen goes away in the form of those bubbles. All right, thank you.